In this presentation, we'll be looking at how biological catalysts, enzymes, speed up chemical reactions. In a previous presentation, we looked at how the energy content of the reactants and products determined whether the reaction would be spontaneous or not. If the reactants are of higher energy than the products, then energy is released during the course of the reaction, and the reaction is spontaneous. However, even though a reaction may be spontaneous, it may happen at a very slow rate. Think of sugar on your kitchen. If you leave that sugar on your counter, it will remain sugar for a very long time, even though the spontaneous reaction that converts it into carbon dioxide and water is exergonic, or spontaneous. In order for a biological organism to take advantage of that sugar, it needs to be able to convert that sugar into useful energy much more quickly than the sugar would convert if left alone. The reactions that we'll be talking about in this presentation are all exergonic. They all happen spontaneously and release energy in the process. When molecules that could potentially react with each other interact, there are two main things that prevent a reaction from happening. The first is that the reactants don't bump into each other with the correct orientation. When this happens, electrons are not able to be transferred in a suitable way, and the molecules bounce away from each other without reacting. The second thing that prevents reactions from occurring is that the reactants may be moving too slowly. If the reactants move too slowly, they don't collide with enough force to disrupt the bonds and make new ones. Again, the result is that the molecules bounce away from each other without reacting. It's only when reactants collide with one another with enough energy and with the correct orientation that a reaction will happen. Now, if you're in a chemistry class, you probably know of a couple of ways that you can speed up any reaction. The first way is simply to increase the concentration of reactants. By increasing the concentration of reactants, you increase the number of collisions that occur. With more collisions occurring, it's more likely that more of those collisions will result in a reaction. Another way to increase the speed of the reaction is to increase the temperature. Remember that the temperature is a measure of the kinetic energy of the molecules. When you increase the temperature, you increase the average speed at which the molecules are traveling, and therefore increase the force with which they collide. Unfortunately, in many biological systems, this second way simply doesn't work, because by increasing the temperature, you would also denature the proteins and otherwise damage the cell. Before we look at exactly how enzymes work, let's look a little bit more deeply at why reactants need enough energy when they collide. This example here is of a spontaneous exergonic reaction. The reactants have higher energy than the products. However, this reaction will rarely occur because the activation energy is so high. In order to react, the reactants must go through a transition state that is at very high energy. In order for a bond between A and B to be formed, the bond between B and C must be broken. However, the reactant BC is very stable, and it's very stable as it is, and breaking the bond will require significant energy. As A collides with BC, the bond must first be broken between B and C in order for the bond between A and B to be formed. Once this transition state has been achieved, energy is released as the bond between A and B is formed, and that AB product becomes more stable. In order for this reaction to happen, A must collide with BC with a lot of force. If the temperature of the reaction is increased, more molecules of A will collide with BC with enough force to overcome that transition state, and the reaction will proceed more quickly. At lower temperatures, however, only rarely will collisions happen with enough force. So, how do enzymes help reactions run faster? Let's look first at the orientation problem. Enzymes are proteins, and as such, remember that they have very special three-dimensional shapes. An enzyme binds two reactants at a particular location on the enzyme called the active site. The active site helps to hold the reactants in a particular orientation, enabling two reactants to be brought together at exactly the correct orientation for the reaction to happen. The reaction, therefore, no longer depends on the reactants colliding randomly in the correct orientation. The second problem involves that high activation energy and having reactants be energetic enough to cross that transition state. Enzymes reduce the activation energy, making it much easier for individual molecules or individual reactions to have enough energy, even at low temperature, to get over that transition state threshold. Notice that the action of the enzyme is to reduce the activation energy only. It doesn't change the free energy of either the reactants or the products. Therefore, the change in free energy, the delta G, remains the same with or without an enzyme. Furthermore, the reaction remains exergonic, with or without that enzyme. Now, biologists used to understand enzyme action according to what has become known as the lock and key model. In this model, the enzyme has an active site that precisely fits the substrate or the reactants. Indeed, enzymes do have very specific shapes that are highly tuned for a particular substrate. In the lock and key model, the substrate binds into the enzyme, and the enzyme causes the reaction, whatever it may be, to occur, and then releases the product of the reaction. Unfortunately, that lock and key model did not adequately explain exactly how enzymes lower the activation energy. A more modern idea involves the induced fit model. In that induced fit model, the enzyme has a shape that fits the substrate, but not perfectly. As the substrate binds to the enzyme changes its shape at the active site slightly to accommodate the substrate. As the substrate binds to the enzyme changes shape, tightening its grip on the substrate. Thus, the substrate fit is induced by the presence of that substrate. When the substrate is bound in the enzyme substrate complex, the enzyme is flexible and stretches the bonds in the substrate, getting the reactants a little bit closer to that activation energy. Additionally, the enzyme actually stabilizes the transition state. The substrate is held by the active site, and this active site may have amino acids that provide a conducive environment for the reaction. For example, they may be able to temporarily donate or remove an electron from the reactants, allowing the transition state to be achieved more easily. In general, we say that the enzyme stabilizes the transition state, therefore reducing the activation energy. So let's summarize how an enzyme works. Initially, substrates enter the active site of the enzyme, and the active site changes shape to accommodate them. The enzyme substrate complex reduces the activation energy, and reactants are converted into product. Once this has happened, the products have a lower affinity for the enzyme, and the enzyme releases them. Once the products of the reaction have been released, the active site of the enzyme is available for new substrates. Now, one very important thing to note here is that the enzyme does not change in the chemical reaction. It's not used up, and it has exactly the same energy at the end as it did before binding the substrates. In order to operate effectively, enzymes often work within a very narrow environment. Let's take a look at this human example in the top graph. The activity of the enzyme increases as the temperature increases. This is for the same reason that all chemical reactions happen at higher temperature. Reactions simply have more energy and are more likely to collide with enough energy, even in the presence of an enzyme, to overcome the barrier of that activation energy. However, when the temperature is much beyond body temperature, the enzyme stops catalyzing the reaction because the enzyme becomes denatured due to the high heat. Remember that when it's denatured, the enzyme is unfolded and unable to carry out its function. Other organisms that live in different environments have enzymes that operate optimally at different temperatures. That is the case with bacteria and archaea that live in hot springs. In order to live at such high temperatures, their enzymes must be capable of not being denatured at those high temperatures. The pH is also a very important consideration for enzyme activity. In order to hold on to a substrate, the amino acids at the active site often transfer electrons or protons to and from the substrate. If the environment is too acidic or too basic, this transfer is disrupted. Our stomach has a very low acidic pH, and so the enzymes there need to be able to operate at those conditions. Lower in our intestines, the pH is much higher, and enzymes in those environments need to be able to operate in a basic environment. Enzymes are very powerful catalysts, and if they're left unchecked in cells, they will catalyze as many reactions as possible, regardless of the needs of the cell. The cell, therefore, needs to exert control over how fast these reactions occur. This control is called regulation of enzyme activity and usually happens through one of two mechanisms. The first mechanism is called competitive inhibition. As you can see in this diagram, the enzyme binds its substrate at its active site in order to facilitate some sort of reaction. In competitive inhibition, an inhibitor, specifically a competitive inhibitor, may also bind at the same site. When the inhibitor is bound to the enzyme, the substrate cannot bind and the reaction cannot proceed. This is called competitive inhibition because the substrate and the inhibitor are competing for the active site. If there are many inhibitors present in the cell, then the activity of the enzyme or the population of the enzymes will be significantly reduced. However, if there are more substrate molecules than inhibitors, the substrate will continue to bind and the enzyme will remain active. The second mechanism for regulating enzymatic activity is called non-competitive inhibition. Another name for it is allosteric inhibition because the inhibitor binds to what is called an allosteric site on the enzyme. The allosteric site is a site that is physically separate from the active site. When an inhibitor is bound to the allosteric site, the enzyme changes shape in such a way that it can no longer bind to the substrate. Therefore, when a non-competitive inhibitor is bound, the enzyme is deactivated. An allosteric inhibitor does not compete with the substrate for the active site on the enzyme, and so the relative concentrations of the enzyme or the inhibitor are not so important. So long as there are enough inhibitors present to bind to most of the enzyme molecules, the reaction will grind to a halt. It's important to note that allosteric regulation can be both inhibitory and stimulatory. In many cases, enzymes oscillate between the active and the inactive forms. An inhibitor will stabilize the inactive form using an allosteric site. An activator, on the other hand, may stabilize the active form and allow the enzymes to spend more of its time in that form, allowing substrates to bind more easily. Now, one very important form of regulation is called feedback inhibition. Feedback inhibition is used to regulate the amount of a product that is produced. In this example, the active site of the enzyme is occupied by threonine, and the result is an intermediate A. After a series of enzymatic reactions, the final product, isoleucine, is produced. The isoleucine, in turn, can act as an inhibitor for that first enzyme. Now, this is a really great system because as the amount of isoleucine builds up in the cell, it will have more and more inhibitory effect on that first enzyme. The end result will be to limit the manufacturing of isoleucine. However, when the amounts of isoleucine in the cell drops, isoleucine will stop inhibiting that first enzyme, and the reaction will start to proceed again. Thus, the cell can maintain very precise amounts of isoleucine in the cell in a very robust way. We'll see this type of feedback regulation play important roles throughout this course.